Non-oxidative phase, it starts with the ribulose 5-phosphate. It is converted to ribose 5-phosphate. Ribulose 5-phosphate is a ketone bearing carbohydrate, and ribose 5-phosphate is aldehydic functional group with 5 carbons, and phosphate group is at the number 5 carbon, and it happens in the presence of phosphopentose isomerase. That's why it's called pentose phosphate pathway, due to this isomerase. In the next step, ribulose 5-phosphate, it is in equilibrium. It's not the steps. It is like ribulose 5-phosphate. It can be converted into this. It can be in equilibrium with this isomer here. This is isomer, and this structure is these are epimers of each other, because this reaction is happening in the presence of phosphopentose epimerase. So xylulose 5-phosphate, it is the epimer of ribulose 5-phosphate, and xylulose 5-phosphate in the next step, along with ribose 5-phosphate, it gets converted into pseudoheptulose 7-phosphate and 3 carbon containing glyceraldehyde 3-phosphate in the presence of transketolase. Pseudoheptulose 7-phosphate and glyceraldehyde 3-phosphate in the presence of transaldolase, they produce fructose 6-phosphate and erythrose 4-phosphate. So these are the same things we studied here, that from ribose 5-phosphate and xylulose 5-phosphate, these 3 carbon, 7 carbon, and these again join together to form 6 carbon bearing fructose and 4 carbon bearing erythrose 4-phosphate. So these are the same thing, just need to remember the names and the enzymes present during the reaction. Xylulose 5-phosphate and erythrose 4-phosphate, they again are in equilibrium, or they produce fructose 6-phosphate. This is 6 carbon bearing compound, and glyceraldehyde 3-phosphate, to keep the stoichiometry calculations balanced in the presence of transketolase enzyme. Oxidative branch is altogether 3 steps bearing apart. First step is glucose 6-phosphate dehydrogenase is committing step. It produces NADPH, 1 carbon dioxide molecule is released, and 1 ribose 5-phosphate is produced. First step is isomerase of ribulose to ribose. Ribose 5-phosphate is the isomer of ribulose 5-phosphate, whereas xylulose is epimerase of ribulose 5-phosphate. Well, this is a little bit it's not complicated, but these are the structures of all this oxidative phase. How glucose 6-phosphate plus Na2 NAD in the presence of 2 NADPs in the presence of water, we do it because it is oxidative phase. In the presence, how it produces ribose 5-phosphate along with 2 molecules of NADPH, carbon dioxide, and hydrogen ion is released. You see, the oxidative part consists of 3 enzymes, and it is responsible for the production of NADPH. In the first step, this one is glucose 6-phosphate, and in the presence of glucose 6-phosphate dehydrogenase, NADP is converted to NADPH, and there is production of 6-phosphogluconolactone, sigma lactone. This is sigma lactone, you read in the last slide. You see, this one is C double bond O, and this oxygen in the form of a ring Now this is a form of a ring, and this functional group is ester. When ester is there in the cycle or in the ring form, then it is called lactone. So in the next phase, the 6-phosphoglucanolactone, it in the form of, in the presence of lactonase enzyme, and with the addition of water, it produces 6-phosphogluconate. There is no more lactone. There is production of carboxylate group at the end. This bond is broken from here. This oxygen goes with this one, and this is 6-phosphogluconate. And 6-phosphogluconate, in the presence of 6-phosphogluconate dehydrogenase, this 6-phosphogluconate is converted into ribulose 5-phosphate, where carbon dioxide is released, and you see this 6-carbon compound is converted into 5-carbon compound. This is 1, 2, 3, 4, 5, and phosphate is at the end with the carbon number 5, and this is now ketone ribulose 5-phosphate. It is no more aldehyde, and in the next step it gets converted into phosphopentose. In the presence of phosphopentose isomerase, it gets converted into indole. This is a kind of an intermediate, and intermediate it gets converted into ribose 5-phosphate. Ribulose 5-phosphate, it isomerizes to ribose 5-phosphate. So this is the whole oxidative pathway, and in the next step, these are the structures drawn properly to show you this, all the oxidative pathways for the pentose phosphate pathway. This is the glucose 6-phosphate with the carbon number 6. There is the phosphate group, and this bond is shown in the pink, that is active bond, and from here hydrogen will be released, and this hydrogen will go with the NADPH, and this oxygen will be available to form double bond O. This is kind of ester, and that's why this is called 6-phosphoglucanolactone. Okay, you can make the structures. 6-phosphoglucanolactone, it in the presence of water, it releases H positive ions, and OH is added in the structure in the presence of lactonase enzyme, and this structure, it opens up here, and this O, C double bond O, and O, it goes here, right, and the whole structure, it builds like this, it opens here, and this is again 6-phosphogluconate, but this is open changed carbon phosphate group is with the carbon number 6 here. 6-phosphogluconate, in the presence of 6-phosphogluconate dehydrogenate, it in the presence of NADP plus, it produces NADPH, and again here hydrogen, from where it is coming, this carbon dioxide will be released as CO2 here, pink, and here there is the addition of there is the removal of hydrogen from here, it comes this way, and there is the formation of I guess CH2OH. Bond splits from here. From here hydrogen goes into here, this hydrogen, and this bond is broken, this bond is again broken, and the double bond comes this way, right, and this bond breaks, and this hydrogen goes with the CH to form CH2OH to balance the whole product. And the product name is ribulose 5-phosphate, because this is kind of a ketone. This bond is this functional group is keto functional group, because it is not the double bond O, C double bond O is not at the end, so that's why it is ketone. It's in between two carbons, and along with that carbon dioxide is released. In the non-oxidative branch, this is reversible integranomers, 3, 4, 5, 6 carbon sugars for synthesis of other compounds. They are produced during this phase. Transketolase enzymes are used, trans and aldolases are used during this pathway. Again, this is the overview how these products, these epimers or isomers, they combine, join together to form 3, 4, 5, 6, 7 carbon bearing compounds. These are the steps for non-oxidative pathway of pentose phosphate pathway. You see, start with the ribulose 5-phosphate, that is the ketone functional group here, 5 carbon, and phosphorus at the number 5 carbon in the presence of phosphopentose isomerase. Because of this isomer, the name of the reaction is ribose 5-phosphate is produced. You see, carbon double bond oxygen, it comes at the end, and at the top, maybe you can say, and it is aldehyde functional group. Isomers, these are isomers, different in structure but same in the chemical composition. Ribulose 5, on the other side, ribulose 5-phosphate, in the presence of phosphopentose epimerase, it converts, or it is in equilibrium with another with its epimer, that is xylulose 5-phosphate. Xylulose 5-phosphate is ketone, but the position of the hydroxyl group is different. These are the epimers. Epimer is the structure in which the position of the functional groups is opposite at the carbon number 3. Right, this is carbon number 3 here, hydroxyl group is at the left, here carbon number 3, this one hydroxyl group is on the right side. So these are the this one is the isomer of this structure, this reactant, and this one is the epimerase of this structure, and two different enzymes are participating in these reactions. When these two products, for one isomer and the other epimer, they join together to form in the presence of transketolase enzyme, pseudoheptulose 7-phosphate compound is produced, and the glyceraldehyde 3-phosphate bearing compound is produced. Glyceraldehyde, from the name it is clear there is 3 carbon containing compound. It is aldehyde, this is ketone, and its name is with ULOS, hulose, like ribulose. This is also ketone, this is also ketone, right? When these two structures, glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate, they join in the presence of transaldolase to produce erythrose 4-phosphate 4 carbon bearing compound, aldehyde group, and another ketonic group that bearing cellulose 5-phosphate compound. Again, this is produced. Like this is reversible as well, and carbon 3, 4, 5, 6, 7 carbon containing compounds are produced. Finally, these two, xylulose 5-phosphate and erythrose 4-phosphate, these two, they produce in the presence of transketolase enzyme fructose 6-phosphate this structure. Fructose is again ketonic functional group bearing, and glyceraldehyde 3-phosphate, the same one. This is produced. This is the final product, fructose 6-phosphate and glyceraldehyde 3-phosphate. These are produced.